So now let's address the first of the biological macromolecules that we're going to look at. Now again, you're going to see this slide again and again and again. Repeat, repeat, repeat. We have 4 classes of macromolecules involved in biology: lipids, proteins, carbohydrates, and nucleic acids. The first one that we're going to look at are lipids. And I like to use the little catchphrase for lipids as defining our world, because lipids are the main component of membranes. And membranes are going to actually delineate the cell, the living biochemistry of the cell, from its environment. If we don't have that, we don't have anything. So let's take a look at lipids. Now, lipids are basically fats. So we think about fats and oils. These are lipids. And when you try to mix fats and water, it doesn't work, right? So this is pouring oil into a glass of water, and you get those globules of oil, and ultimately they all clump together, and you get this separation of the aqueous solution from the non-aqueous solution. And this is going to introduce some very, very important concepts when it comes to lipids. This is the idea of hydrophobic and hydrophilic. So hydro means water. So if something is labeled hydrophobic, like a phobia, a fear, it is water-fearing. If something is hydrophilic, philicity, loving, it is water-loving. Hydrophobic, water-fearing. Hydrophilic, water-loving. So hydrophobic molecules generally have no charge or polarity. Right? So remember we talked about hydrocarbon backbones. What we're going to see is that most lipids are mostly hydrocarbon. So they don't have a charge or polarity. They tend to clump together in water. This is greasy stuff, right? Because water has charge, this partial charge in every water molecule, charge wants to find the opposite charge. So when you present a water molecule with a lipid, with a hydrophobic molecule, it doesn't find any plus or minus to interact with, and it really wants to interact with a plus and minus. Hydrophilic molecules, on the other hand, are going to have charge or polarity. They're going to readily mix with the polar water molecules because they have some polarity too. They have some positive or negative that water wants to interact with, right? So remember, every charge is always looking for its opposite. And the forces of the universe of physics are driving those positives and those negatives together. There's that attraction, all right? So lipids are hydrophobic. When we look at them, we can see that, in fact, they are mostly hydrocarbons. Again, hydrocarbons are going to be almost completely nonpolar bonds, right? The hydrogen and the carbon have almost exactly the same electronegativity. Because of that, they're going to share those electrons almost exactly equally, and hence there's no polarity there. So if I take these fatty acids and link them together with a glycerol, I get what we think of as basically a fat molecule, okay, a triacylglycerol. It's a lot of carbon and hydrogen. It's a lot of non-polarity, all right? So this does not mix well with water. So this is now what we think of as butter, animal fats, okay, or vegetable oils. Now, there's a lot more to lipids than what we're talking about just in this short introduction class. So you can talk about saturated versus unsaturated fats. What I recommend is exploring a nutrition class to learn more deeply about these aspects of lipids and fats. For now, we're going to talk about fats as being a biological function for energy storage. So that's the fat that we eat or the oil that we derive from vegetables. It's a storage molecule because all those hydrogen-carbon linkage, all those carbons in a row, actually is a great way to store a lot of energy. So if you grab any piece of food that you buy in the store, you'll see on the back fats, you get 9 kilocalories ultimately per gram, whereas proteins and carbohydrates only give you 4. So fat is actually really, really rich in energy, and it's used biologically for storage. That's one of its purposes. The other purpose that lipids or fats really serve is, like I said before, making membranes. And the specific kind of lipid that I want you to know is called a phospholipid. Now, we've seen this picture before when we started introducing the idea that all molecules that make up cells are made of atoms. So we've seen this picture before, and now we can see all these atoms linked together, carbons and hydrogens and some oxygen in there, and they actually see a phosphate. But what you'll notice about this particular type of lipid, this phospholipid, is that all the functional groups, all the charge, all the oxygen is on one end of the molecule. And what this creates is a hydrophobic portion of the molecule where the fatty acids are, the hydrophobic tails, and a hydrophilic, what we call head group. So the head of this molecule actually wants to interact with water. Why? Because there's charge there. Look at the positive charge on that nitrogen up top, the negative charge on the oxygens around the phosphorus. Right? That's called a phosphate group. So now you've got a molecule, and the fancy word for this is amphipathic, which you don't have to know, but this molecule that has one end that wants to be with water and one end that wants to hide away from water because it doesn't offer any charge is very special because it will spontaneously form structures. Now those structures are either a micelle or what we see as a phospholipid bilayer. And this is a beautiful structure because when you look at it, using the idea of that cartoon that the gray circles are the hydrophilic head and the little tails are the hydrophobic fatty acids, you can see that all the hydrophobic tails are hiding away from the water because they're facing to the interior of this bilayer, and all the hydrophilic head groups with their charge are out interacting with the water. This is a membrane. Now I have a barrier that I can establish chemistry going on inside or outside of the cell. As we again look, this makes up the cell membrane, right? So this barrier isn't necessarily very physically robust, but chemically it's very robust because anything that has to move through it has to go through that greasy layer. And anything that is in the environment, basically around these cells, must be an aqueous solution. So it must be interacting with water to a certain extent. And so it doesn't want to cross that hydrophobic layer. All right? So that's how membranes work. And the foundation of membranes is that phospholipid. So I definitely want you to understand that. A little bit of charge gives you a hydrophilic head. With the fatty acid tails, gives you a hydrophobic tail. All right?